as we knew it around the globe, the degree of uncertainty and anxiety about our future has never been greater. At the same time, our resolve to defeat this pandemic is even stronger, which I'm certain will ultimately help humanity prevail. When the COVID-19 pandemic was rapidly accelerating in early March, our university community unified to bring our scientific expertise and resources to launch an attack against this pandemic. The actions by our university research community is too numerous to present all of them here, but a complete list is available on our website, VPR website. Contributions include rapid development of testing capabilities, models to predict the course of the pandemic from an epidemiological point of view, testing potential drugs for cure, vaccine development for prevention, and novel approaches to meeting the challenges of PPE shortages, swabs, and much more. In this panel, we will have examples of such efforts. You will directly hear from the people doing the work. Before we start, please note that we're not using the chat feature. Several questions were emailed to us in advance. We will try to answer some of them following the panel's presentations. If you have any questions you want to submit, please write to askresearch@virginia.edu, and we will try and address them after the webinar. A recording of this will be available afterwards on our website, research.virginia.edu, and the program is currently being streamed on our YouTube page. This event is being recorded and can be found on our website later on. Without any delay, let me introduce our panelists. First, we have Dr. Madhav Marathe. He's an endowed distinguished professor in biocomplexity, director of the Network System Science and Advanced Computing Division of the Biocomplexity Institute and Initiative, and a tenured professor of computer science in the School of Engineering at the University of Virginia. His areas of expertise are network science, artificial intelligence, high performance computing, computational epidemiology, biological and social coupled systems, and data analytics. He is currently coordinating the Institute's modeling and analysis efforts related to COVID-19 pandemic. The modeling group is supporting the state of Virginia and other federal agencies in their planning and response efforts to control COVID-19 outbreak. Dr. Marathe will talk about his pandemic modeling work and how it has helped with the preparations from a public health and epidemiology point of view. Dr. Amy Mathers, Associate Professor of Medicine and Pathology, Division of Infectious Diseases and International Health at UVA. Her expertise in tracking bacterial DNA sequences has advanced the understanding of where antibiotic resistance pathogens originate and how they spread in even the most sterile places like the hospitals. Dr. Mathers will talk about her efforts in developing a test for COVID-19 at UVA and how this is helping the region and the Commonwealth. Dr. Patrick Jackson, Assistant Professor of Medicine, Infectious Diseases, and International Health. He provides treatment, counseling, and education for patients with HIV and AIDS in the UVA Ryan White HIV Clinic, and provides general infectious diseases care. He's a virologist in the UVA Miles H. Thaler Center for AIDS and Human Retrovirus Research. He is the principal investigator for a clinical trial for using remdesivir to treat patients with COVID-19. He will be talking with us about his work on treatment development for COVID-19. And finally, Dr. Stephen Zeichner will speak. Dr. Zeichner is a professor of pediatrics who studies infectious diseases, disease pathogenesis, and works to develop new therapies and diagnostics for infectious diseases and cancers. He is working on a vaccine for COVID-19. He will be sharing this information with us today on his approach to developing a vaccine. Let's start the panel. Let's hear from Dr. Marathe. The format of this panel is each panelist will speak for about 10 minutes, and then we will have we have received the questions, and I will be moderating, asking the questions and do a Q&A. Dr. Marathe first. You may be muted.
Yeah, can you hear me now?
Yep.
Okay. Thank you very much, Ram, and your team. Privileged to speak today at the UVA panel. Before I go forward, I want to just say that I would like to dedicate this talk to the amazing efforts by our folks, the public health community, and also to Dr. Robert May, who is a pioneer in epidemiology and really is the founding father of modern computational epidemiology. So let me begin. The work I'm going to present is not my work by any means. It's the work that our team has been doing together as a group for over 2 decades now, starting back when we were in Los Alamos, and has also been funded by a number of different grants that I have listed here. Um, so quickly, I would like to give a short 2-minute tutorial uh, on COVID because I'm the first speaker and it'll set the stage. I don't think so anybody needs to be told how it started back in China, but nevertheless, just to remind all of us, it's a zoonotic disease, and that meant that we were all susceptible to this particular disease. It started around December 2019 and quickly became a pandemic, as we know right now. It's caused uh, significant damage and probably the largest epidemics that we have seen since the 1918 influenza pandemic. Uh, I think it's important from an epidemiological perspective to say why this became a pandemic potentially. One, as I said, all of us are susceptible. It's a zoonotic disease that jumped to humans very recently. But there are other important reasons as well. One that includes urban spread. It started in a part of China that was very densely populated. This particular disease has a property that it has got silent spreaders. So people are asymptomatic and yet they can be transmitters of the disease. That makes it very hard to control. Third reason, in my opinion, that is important is China was very well connected to the world. We participated early on in other outbreaks, but the connection China has to the world in terms of business and mobility made this very hard to control. And finally, because it's a zoonotic disease that has just jumped, we don't have a pharmaceutical intervention. And Amy and others are going to talk about it a lot as we go along. So from an epidemiological perspective, as of today, really the only solution we have is social distancing. So our institute has been working on this problem for a long time. We were prepared for this in some ways. We have a long-standing expertise, as I said. We began tracking this in 2019 December, sometime middle of December or late December, and formally began work in January. Our group has about 80 staff scientists, students at UVA, as well as our collaborating institutions, and been supported by a number of agencies in doing the work, but we also support them in their analytical efforts, notably the DoD, the CDC, our own state, and the local community here. And I'll say a little bit about the advances that the work has led to as I go along. So you all know the way this works in mathematical epidemiology. The standard way is to build what are called compartmental mass action models, and they're quite useful, but it turns out that in this particular setting, those models, for all their strengths, also have a few shortcomings maybe, or maybe that's too strong a word, but certainly things that you would want to do with the models that those models can't do very well. So our group has taken a fairly unique approach to tackling this particular epidemic outbreak. And the way we do this can be best described in 3 simple steps. If you want to understand how disease spreads in a city like Charlottesville, for instance, then our first step is to build what we call a digital twin of Charlottesville. So you build a computer representation of Charlottesville, the individuals who live in it, the roads, the buildings, the kind of activities people do, where they go around during a normative day, to essentially get a social contact network, a network on which diseases such as coronavirus, but others would spread. This particular network captures the interaction aspect of it. The key aspect of this digital twin is it represents Charlottesville quite well, but preserves the anonymity and privacy of individuals. So you will not find me in this, for instance, but you would find somebody like me. And that realistic representation allows us to build interesting models. The second step is to build high performance computing based simulations of how diseases would propagate over such networks. These simulations are critical. That's the only way we can study dynamics over such large networks, and are then used in the third step to carry out what I call epidemiological workflows, things that range from calibration to forecasting to counterfactual analysis to resource allocation and inference. So the kinds of problems we have studied in the last almost 4 months now range from questions such as importation of disease to from an originating country to various parts of the world. We then moved on to projections, forecasting basically the course of the pandemic with and without interventions, counterfactual analysis, economic impact analysis, resource augmentation, such as building new field hospitals, resource sharing, such as sharing ventilators and other precious resources that are in short supply, and finally projecting the demand on the healthcare system, which is very important right now. I'll quickly show you a couple of different dashboards that we have built. All of this is in slides that will be made available, but these dashboards have proved very useful. One that is going to be released soon is on demand projection. One that has been already released is a surveillance dashboard that has been very, very popular in the community. VDH uses the data that we produce and in their dashboard as well. And finally, there is a system that we've been working with Stanford to collect all sorts of social distancing measures that local governments and the state governments are putting together. And we hope that this can help us drive the epidemiological modeling at the county level. So the kinds of advances that we have had to make in trying to address the challenges range from building very detailed social contact networks. We started with building small regions, moved to building the entire US, and now we have very early versions of a global social contact network that we think is going to be very useful in the coming months. We've also scaled up our epidemiological simulation models, again, from small systems to now the ability to simulate 300 million node networks with about 8 billion edges in it. We have created pipelines that run on supercomputers, such as the one at Pittsburgh Supercomputing Center and our own Rivanna computing cluster here. We've been using these clusters every day now for last, I would say, 6 to 8 weeks through the night, and 20,000 cores have been made available to us thanks to the Exceed and the Pittsburgh Supercomputing project. And finally, we've developed an interesting set of machine learning techniques for calibration, forecasting, and inference analysis. So let me close quickly by just telling you the kinds of things we are doing for our Commonwealth. About 6 weeks back, we were asked to help the Commonwealth understand the impact of COVID-19 on the state of Virginia. And one of the things we've been doing is to project forward the case count across the state in the kinds of things we would expect from the epidemic dynamics that are evolving rapidly. And we've been using the kinds of model I've told you. And the outcomes that we're measuring include the number of infected individuals, hospitalizations, geographic spread, which is very important. So our models are able to predict not just at the state level, but every county within the state. And they're data driven, which means we take input from the state VDH dashboard at a weekly basis and then use them to drive our models forward. Just as one example here, I've shown you 2 plots, the kinds of plots we share with them every week, which shows you how we think that the epidemic is going to essentially take the next, project itself in the next few weeks or so. The black line shows the real numbers that are obtained from VDH, and all other lines effectively show you what is likely to happen under different scenarios. For this talk, I think it's best to understand that these scenarios capture our understanding of level of social distancing that is ongoing. The bottom line is, at the current moment, we think that the social distancing efforts are working, although our models suggest that their effects might be waning. Many states have started reopening their workplaces and restaurants and other places. This is certainly going to have an impact on Virginia as well, and we believe that this might lead to a somewhat increased incidence rate in the coming weeks. The second thing that we think is important to note here is that the hospital capacity is okay right now. We don't think we're going to run out of capacity. And I've been very careful in not showing you peak curves and when the peak would occur, not because it's not important, but I think it's important to note that those things are really mathematical abstractions. The goal of the state, all the epidemiologists in the state and the policymakers is to make sure that we never hit these mathematical abstractions at all and keep the curve to a level that we can manage. So I will stop there by simply saying that models indeed can be useful. I know a famous quote by Box that all of you have heard by now, but I do believe models can indeed be useful, and real-time epidemic science is possible. And finally, I want to conclude by saying that models go beyond being useful just for prediction. They really allow us to understand what's happening and help us do a number of different tasks that policymakers are interested in. So with that, I will stop and we can take questions later, I guess, Ram, as I understand.
Thank you, Madhav, for that presentation on your modeling efforts. Next, I invite Dr. Mathers to talk about testing capability development at UVA. Amy?
Thanks. Let me, can you see my screen? Great.
Yes.
So thank you so much for the opportunity to speak to everybody today about just some of the testing. By way of background, I'm not a viral molecular diagnostic person, but I am definitely in diagnostics in the clinical lab, and that's where some of my research is as well. So I thought I'd just spend a little bit of time on this slide to kind of just kind of walk through the story of testing and why it's important and why it's been such a focus for the COVID crisis, and then where some of the hurdles have been. So I put this slide up, and in the yellow boxes are sort of the things that we did, and then the blue boxes are more what was happening in the world at the time. I will say that because of the way the arrows light out, they're not all quite to scale. Like there's things that are happening that actually happened a little bit earlier, but I tried to kind of put out a timeline. And what's really important is on this timeline, first off, there's not really a yellow box until mid to late February, because just so you know, in a clinical lab, generally, we try not to develop our own tests in a clinical lab, and because it makes a lot more sense to develop an FDA cleared commercial test and use those platforms, because those can allow a lot more high throughput. So, but come mid-February, it was looking like, that was after the first case was January 21st in the United States. And then it was looking like mid-February, there was going to be ongoing transmission in the US, and we were really going to have something. But the problem was simultaneous with us recognizing that we needed to have a testing in-house, the FDA got in the middle of the testing, the testing environment in the US, and it made it so if we wanted to develop our own test in-house, we would have to clear it with the FDA. Normally, as a clinical lab, we do not have to clear any test through the FDA. We adhere to good practices. It means if you're doing your own test that's not FDA cleared in your lab, under your CLIA license, you use a lot of controls, and it's still a valid test, and you report it as a lab developed test. And so that would have been the way that we would normally go. But because the FDA got in the middle of it and said, nope, you're going to have to clear any lab developed test through the FDA, we really, really did not want to have to file FDA paperwork because we'd never done it before, and it's a really big ask for a clinical lab. So with all that in mind, we really wanted to stick with the CDC protocol, because if we used the exact CDC protocol, then we wouldn't have to file with the FDA. So all of that to say, what ended up happening was when you went to go use the CDC protocol, you couldn't get any of the reagents because everybody else had the exact same idea we did. And so you could not get any of the specific things you needed for running the CDC protocol in your own lab. What we did at UVA that sort of made the press was that we got the FDA to allow a lot of different platforms, different reagents to be allowed under the CDC umbrella without filing a full protocol with the FDA. And so when they say it was a CDC developed test, we didn't really develop much of anything except get allowances from the FDA. And I'm going to talk a little bit about how we actually did that here and some of the amazing stuff that researchers around campus did to help us support that. It was pretty awful in the early weeks of March when we could not get a test cleared because we couldn't get reagents, because we couldn't get any of the materials we needed, whether it was primers and probes, control virus, a platform, extraction. All of that was back ordered and we couldn't get it. So what was happening was people were getting admitted to the hospital and basically had to be presumed to be positive with COVID, and you couldn't test them to say, nope, they're not positive. So the day that commercial testing opened up in the country there around March 10th, it was basically LabCorp and Quest, and they had a capacity of 10,000 tests a day roughly. And essentially their capacity got overwhelmed by the entire East Coast such that their turnaround times went as far out as 11 days. So that wasn't helpful to our inpatients. And so as soon as we got a test available on March 18th, we immediately pivoted and started testing not just for our own patients, but for other patients. But that was on the CDC platform that was very, very manual.
Connected to that.
As soon as we possibly could, we got commercial testing. It was like 2 days later. As soon as there was a commercial product we could use, we got that up and running. And then we've got a third and a fourth commercial product all up and running. We've turned some commercial products away because they don't perform very well. And it is a little bit of the Wild West with what's going through clinical labs right now. I'll talk at the very end about the swab shortages. So what's really needed, and just briefly to kind of go through this, what's needed for a COVID test? First off, you need a flocked swab ideally, which is nylon and inert and grabs that virus and keeps it. It's an RNA virus, so somewhat delicate. We then want to put that in a viral transport media. And again, I'll talk at the end about sort of the supply chain here. You need enough PPE and whatnot to collect from a symptomatic patient that you have to presume might be positive for coronavirus. Put a swab back through their nasopharynx or throat. There are other things on the market that are coming that you might have heard about in the news. You need to then get it to the lab in our current form. Maybe that'll change if they have in-home testing. Some of that stuff's not performing all that well. And then we have to safely in a BSL-2 hood get the virus out, process the specimen, get it into viral inactivation, put it on one of several platforms, and then look at what the PCR results are, or whatever the diagnostic platform is using, and then relay those results. So that's kind of the nuts and bolts of testing. And I'm just gonna finish with that kind of background, an understanding around what happened in the early days. So when we say we developed the UVA test, what we really did was, because you couldn't get any of these ABI instruments, they're back ordered by still 8 weeks. And people that were trying to get them could not get these platforms. And so what I did was I just went to the finance people and got a list of every lab that had one of these. And I just went door to door to labs asking if we could take their equipment. And generous people like Melissa Kendall and James Casanova lent and still have lent their pieces of equipment to making COVID testing happen. This is Melissa Kendall reminding me that this was her first piece of research equipment that she ever owned. And so we should all give a big thanks to her for donating that. We took equipment from Chris Musculloch, as well as from his lab, as well as a lot of reagents that were donated from the university. It was just a really warm outpouring of people supporting us getting this testing up and running. As soon as we could, we quickly pivoted to commercial products, although we still have to use the, the research platform for the lower respiratory, because none of the lower respiratory, like a bronch or a lower respiratory sputum sample, are validated on any platforms other than the first CDC platform. So now we can do platforms that have 96 isolates at a time you can do, or specimens at a time. And then this one is a 45-minute on demand. It says TB PCR, but that's because I didn't have a picture. On demand test, and it takes 45 minutes to say COVID, present, absent. The last thing I'll finish with is this sort of research put together. I've worked with Will Guilford for many years around mostly sink and environmental and building things around that. And we partnered on trying to make swabs, which have been desperately short. So using prototypes of things that were on the market, we basically have not 3D printed, because again, 3D printing, they're fairly brittle, and these are quite delicate, the swabs. You don't want them breaking off in someone's nose. And so we're using polypropylene, and then we have them flocked with the nylon flocking at the end. And I'm in the middle of trying to wrap up an FDA trial to get those cleared so that we can distribute them around the state. And hopefully UVA will be manufacturing about 60,000 a week and distributing to others in the state. The viral transport media has also been very short, and Larry Lum has donated his lab GMP facility to making viral transport media for multiple people, including the Department of Health, to get this testing done. That's all I had. Thanks.
Yeah, thank you, Amy, for that very detailed and account of how these things happened. And I'm sure there'll be questions we can discuss. Thank you. Next, we have Dr. Jackson, who will be talking about his treatment development for COVID-19. Patrick?
Okay, sorry, technical difficulties. All right, thanks. So I'll be chatting about the clinical trials that were being conducted at UVA for COVID-19 therapies. So, so you may have seen this news report, and it's not often that clinical trial in virology is considered to be breaking news. But this is a report of Dr. Anthony Fauci talking about some early results of a trial for a drug called remdesivir. Oh, you know, I think. Oh, sorry. I think I just ended my slideshow. There we go. So the study that Dr. Fauci was talking about led to the FDA rapidly issuing an emergency use authorization for remdesivir to be used in the treatment of COVID-19. And at UVA, we participated in this trial that led to that new therapy being so rapidly rolled out. So I'd like to talk a little bit about why we decided to get involved in that trial, how we were able to contribute to generate that important data, and also where we're going from here. So in COVID-19, as in other infectious diseases, the damage that occurs in the body is thought to be due to a combination, both of replication of the pathogen, which in this case is the virus SARS-CoV-2, as well as the inflammatory response of the immune system. So while the immune response is necessary to control the growth of the pathogen, when it becomes too robust, it can actually damage some of their surrounding tissues. Okay. So it can actually damage some of the surrounding tissues. So when you're thinking about drugs for the treatment of COVID-19, those drugs can really be categorized in two ways. We can be thinking about drugs that primarily affect viral replication, or we can be looking at drugs that primarily modulate the response of the immune system. In the category of drugs that primarily affect viral replication, these include things like remdesivir, favipiravir, chloroquine and hydroxychloroquine, the latter of which is marketed under the name Plaquenil, lopinavir, ritonavir, which are two HIV medications marketed under the name Kaletra, and ribavirin. Under the category of drugs that primarily impact the immune system, these include mesenchymal stem cells, baricitinib, the IL-6 inhibitors that include tocilizumab and sarilumab, anakinra, and corticosteroids. And then there are a few drugs that kind of bridge the gap between the two, and these include things like convalescent plasma and interferon. Here at UVA, as mentioned, we've been part of the trial of remdesivir. And the reason why we wanted to get involved in that is that it's a very promising sort of drug for this purpose. So remdesivir has been around for about 10 years. It's a nucleoside analog that has broad spectrum activity against the RNA viruses. And the way that it works in general is that it interrupts the process of an RNA virus replicating its own genome. So it interrupts a vital part of the viral life cycle. We know from in vitro experiments and from animal models that remdesivir is effective against multiple coronaviruses, including SARS-CoV, MERS-CoV, and SARS-CoV-2. And while remdesivir had never been used previously in clinical trials for coronavirus, it was used for clinical trials for Ebola virus disease in West Africa and the Democratic Republic of Congo. While remdesivir was not the most promising active agent used in those trials, we were able to get information suggesting that it's a relatively safe drug to use in humans. So as a result of the promising preclinical data suggesting that remdesivir would be effective for treating SARS-CoV-2, and the clinical trial data suggesting that it's a safe drug to be used in patients, 5 clinical trials were launched worldwide, including 3 in the United States. Of the clinical trials in the US, 2 were sponsored by the manufacturer of remdesivir, Gilead Sciences, and 1 was sponsored by the National Institutes of Health, and the NIH trial is the one that UVA has been participating in. The UVA clinical trial is called the Adaptive COVID-19 Treatment Trial, or ACT-1. And so this includes hospitalized patients who have mild through severe COVID-19. So we're really able to look at the full spectrum of illness. We were including patients who were hospitalized but not even requiring supplemental oxygen, so not very sick at all, all the way through patients who were severely or critically ill with COVID-19, including patients requiring mechanical ventilation or even ECMO. People who elected to participate in the trial were randomized to receive either remdesivir or placebo through an IV for up to 10 days while they were in the hospital, essentially a coin flip about whether a patient received remdesivir or a placebo. And then we looked at the time to clinical recovery within the first 28 days after randomization. And here we defined clinical recovery as being a patient being well enough to either leave the hospital or a patient whose clinical disease had entirely resolved, and if they were still in the hospital, it was purely for infection control purposes. 1,063 patients were enrolled worldwide at 68 sites. UVA was one of only 2 sites in the state of Virginia, and we were attractive to the NIH as a site in large part because of the amazing testing work that Dr. Mathers was just speaking about that allowed us to make the diagnosis of COVID-19 so quickly for our patients. The data that I have to share about ACT-1 is based on a preliminary analysis by the Data Safety Monitoring Board. So every clinical trial has a DSMB, that's an independent body overseeing the trial, that takes a look at the data periodically to make sure, on the one hand, that there's not harm that's happening to patients that would mean that the trial needs to stop early. And on the other hand, that there's not such a fantastically positive result that the trial shouldn't stop early so that new treatments can get rolled out. The DSMB in this case took a look at the data after all 1,063 patients had been enrolled in the study, but only 606 patients had reached that preliminary endpoint. Another 400 odd patients are still part of the trial. So in this analysis does not include data from them. The DSMB found that patients who received remdesivir recovered in about 11 days, while patients who received placebo took 15 days to have clinical recovery. This is a difference that's statistically significant and also quite significant from the standpoint of a clinical side of things, because we know that COVID-19 takes a long time for patients to recover. So shortening that gets patients out of the hospital faster. And being able to move patients back home also decreases the stress on healthcare systems, especially in parts of the world where there is more burden of disease from COVID-19 than we've observed so far in Charlottesville. The DSMB also found that in patients who received remdesivir, 8% of those patients died, whereas 11.6% of patients who received a placebo died. Obviously, that's highly clinically significant if that difference is true, but that difference didn't quite reach the level of statistical significance. So it'll be important to follow up when we have complete data from this trial available to see if that difference in mortality is a true phenomenon. So it's based on this data that the FDA issued that emergency use authorization. And that's going to allow physicians in the United States to prescribe remdesivir for their hospitalized patients with COVID-19 who have pulmonary symptoms, even outside of a clinical trial. So this is a remarkably fast development of an antiviral drug, which is a classically very difficult sort of thing to do, and a very impressive result to get really high quality information for a randomized controlled trial in the midst of a pandemic. Now, right now, there are difficulties in distributing remdesivir. The federal government is in charge of that at the moment, and we are working hard at UVA to get access to that drug so that we could use that for our patients here. We are continuing to do research on clinical treatments for COVID-19 here at UVA. So Act 1 is moving directly into a second phase called Act 2, and we're going to be looking at the combination of remdesivir and a drug called baricitinib, which is a JAK1, JAK2 inhibitor. The idea here is to see whether the combination of an antiviral drug along with an immune modulatory drug can yield better outcomes for patients than using either class alone. Another group here at UVA is currently looking at mesenchymal stem cell therapy for patients requiring mechanical ventilation with COVID-19. The idea here is that patients can receive adult stem cells infused into their bodies that will hopefully take up residence in the lungs and then tamp down on inflammation locally and help protect lung tissue from that robust inflammatory response. And finally, another group is currently actively recruiting patients looking at convalescent plasma. The idea here is that patients who have COVID-19 and recover on their own will oftentimes develop useful antibodies in their blood. If we can take those antibodies from recovered patients and give them to patients who are currently sick with COVID-19, then that convalescent plasma may help to bind up the virus and may also help to reduce inflammation. So none of these trials would be possible without a large number of people working together. Most importantly, the participants have given generously of their time and efforts and allowed us to learn more about COVID-19 and how best to treat people. And there's a huge number of people who have worked directly on these trials, not all of whom have pictures that I was able to find, but our clinical research coordinators and pharmacists, Miranda West, chief among them, have been fantastic. Frontline nurses and physicians taking care of these patients, and other study co-investigators, particularly Dr. Tyson Bell. Happy to answer questions about this trial after the conclusion of the talks.
Okay, thank you very much, Dr. Jackson, for the talk and explaining to us how UVA played a part in laying the first building block for a treatment against this virus. Next, we will have Dr. Stephen Zeichner talking about his work towards finding a vaccine. Dr. Zeichner.
Thanks, Ram, very much for the invitation to talk to the group. I'm going to talk about our effort to develop a vaccine for COVID-19. First, I need to thank the folks that are doing the actual work, my postdoc, Denny Maeda, and technician Vignesh Rajasekaran, Hannah Yu, and our brand new technician who just started today, Nakul Dar, who's taking Vignesh's place when he goes to medical school this summer. We have ongoing collaborations with Mark Kester to do some follow-on technologies from what I'm going to talk to you about involving mini cells. And we have a great partner on the other side of the Blue Ridge, XJ Meng at Virginia Tech for some exciting animal models. And thanks to you, Ram, and the other folks in the UVA administration for making this all possible. There are published accounts of something like 200 plus vaccine development efforts. I think many of these vaccine development efforts focus on finding a really cool technology, showing that it can work, and then worrying about how to make it at scale in a globally appropriate fashion later. We took a kind of complementary approach where we said, look, what are the design goals for a vaccine that we can give to everyone in the world? And then how can we come up with something that meets those design goals? So I think for a good vaccine, what we want is something that's safe and effective, of course, but also globally appropriate. And by that, I mean inexpensive to produce, ideally less than $1 a dose. Because if you have a country where the annual GDP per capita is $1,000 per person, a $100 vaccine is going to be worthless. We'd also like to use something that employs a well-established technology that has previously yielded approved vaccines, something that we can produce rapidly and that has a production process that's forgiving. We also want something that's logistically friendly. So in other words, a production process that can be scalable, that uses feedstocks that are inexpensive and readily available, and ideally no need for a new factory. If you need to build $1 billion factory, the pandemic could be over by the time you get your building permit. And something that's stable with a minimal cold chain requirement and resistant to evolution of resistance. The group at Los Alamos has started to see some evidence of mutations that may confer some additional spreading capabilities. It's a little controversial now, but we want a vaccine that is going to work not just now, but for the future in case the virus evolves. And I think it would be good to have a vaccine that targets immunity to a particular part of the virus. That means that we could develop an assay that could distinguish between someone who made an immune response to the virus, to the infection, and someone who made an immune response to the vaccine. So the technology that we're working on is a killed whole cell bacterial vaccine that puts proteins from the virus on the surface. Now, you might think killed whole cell vaccines, well, that sounds a little weird, but in fact, they have been in existence for more than 100 years. And there are several killed whole cell vaccines that are currently in production, approved by various licensing agencies. And in the case of these killed whole cell cholera vaccines, stockpiled by the WHO around the world, waiting for the next cholera outbreak. And these are in fact amazingly inexpensive to produce. So the company that makes this, Uvicol, U Biologics in South Korea, they wrote a paper a year or so ago that said that from a single 100-liter fermenter, they made, which is the size of the gas tank in your car, they made 6 million doses in 1 year at $1 a dose. So you can imagine this would be highly scalable. And in fact, many middle-income countries already make killed whole cell vaccines or derivatives thereof. So just a little bit of background. We had been working on a technology because we were interested in making an HIV vaccine. And on the left here, you see a picture, artist rendition of HIV, and on the right, the famous picture of SARS-CoV-2. And these are both, they're different viruses, but they all have this class 1 fusion protein on their surface, a trimer, that mediates the fusion of the virus envelope to the host cell, enabling to the virus to infect the cell. And the HIV vaccine field has coalesced around a strategy in which patients who have been infected with HIV are screened. The immunologists have looked to see which of those patients make good immune responses to the virus. They make what are called monoclonal antibodies from those patients and then map where those monoclonal antibodies bind. And one of the great sites is this fusion peptide, a small 10 or 15 amino acid sequence that actually mediates the fusion of the virus to the new host cell. And because they're both class 1 fusion proteins, SARS-CoV-2 also has a fusion peptide, and this is the amino acid sequence down below. So we said, well, let's just try to do the same thing for COVID-19. Here on the left is a recent structure of the SARS-CoV-2 spike protein. And you can see this is the 15 amino acid fusion peptide that lies just tucked in over here. And so that's what we're targeting. Now, the way we're doing this is we're using a class of proteins that gram-negative bacteria like E. coli have called autotransporters. And these are one way that the bacteria put thousands of proteins onto their surface. The bacteria need to do this to interact with their environment. But this protein is special in that it has part of it goes through the inner membrane of the bacteria, and another part of it over here makes a little pore in the external membrane of the bacteria. And then there's this passenger protein that flips through to be displayed to the outside world. So using genetic engineering, we can put a DNA sequence in here in place of the native passenger protein DNA sequence and get this thing to put 200,000 copies of the protein that we care about, in this case, the vaccine antigen, on the surface of every E. coli. So we synthesize what's called a plasmid that enables us to put this into bacteria and have the bacteria put it on the surface. And when we did that with an HIV antigen that we got from our collaborator, Peter Kwong at the Vaccine Research Center at the NIH, we can see that we can show that there's binding to that HIV protein. And then when we immunize mice, we found that the mice actually make sera that can immunize, that can neutralize, inactivate HIV, which was exciting, but we wanted to make it better. So we were thinking, well, we know we want to put a particular protein on the surface of the bacteria. The bacteria doesn't know that, and the immune system doesn't know that that's the protein that we care about. So what would happen if we took away many of the proteins on the surface of the bacteria? And there's a group in Japan at Tokyo Metropolitan University that was asking the question, what does it take to make a living bacteria? And they systematically removed more and more and more of the bacteria's genes until they got up to about a 30% genome reduced bacteria and still had a living bacteria. Well, when we got those cells from those from the group in Tokyo, we found that when we put our HIV fusion peptide in there, it bound much, much better, which we think means that that's going to be a much more effective vaccine. So here's the platform that we come up with. We have somebody identifies an antigen. We synthesize the DNA for that. We clone it into our plasmid. We transform it into the bacteria. We optimize expression, inactivate it, and then have a vaccine that we can test. And when things are humming, we think we can do this in about 3 weeks. So we said, of course, when the COVID-19 pandemic became a problem, we said, let's try it for COVID-19. So we synthesized these DNAs as a single one and as 5 strung together. We cloned it into our system. And now we're going to optimize the expression of the viral antigen, the fusion peptide, make formalin inactivated vaccines, immunize animals, test for antibody production and for the ability to neutralize the virus, and then to protect mice and pigs in challenge experiments. And if all that works, we'll be on to clinical development, which we hope can happen very soon. And that's it. Hey, have you guys been hearing me?
Okay, yes, yes.
Okay.
We're just waiting, want to be sure that you're finished before.
Yeah, no, sorry,
Okay.
I should have said the end or something.
No. I knew this was the last slide, but I just. Yeah, well, thank you so much, Stephen, for that very, very hopeful and optimistic and very sort of a promising approach to vaccine development. We have about 10 minutes, and we had, like I said, we had a bunch of questions. And first of all, I want to say that as Patrick said, it is a collaboration, and it's Amy's work, is helping Patrick get into the trial, and we are playing a major role in the national trial that actually led to the first, building block using the remdesivir trial, and the work of Madhav leading to, a guidance, a predictive a guidance as we go along. As data comes along, his models get stronger, but for lack of information, really providing that light to move forward and make decisions. So really, it's amazing work. So let me talk about, ask a few questions that have come into play. Maybe let me start with Madhav. From an epidemiological perspective, in the immediate term, what are the behavioral elements that work towards controlling the spread of COVID-19?
Yeah, it's a good question, Ram. I think really the only strategy we have right now to control this is social distancing. And the good news is that although it seems harsh, there are multiple ways to do social distancing. And I think a couple of interesting technologies would help us a lot. I think the work that was described by Amy and others in terms of tests would be very crucial. But I think there are multiple other ways where social distancing can be combined with testing and contact tracing through either human contact tracers or digital devices-based contact tracers that could, in our opinion, reduce the burden of this disease. There is no other way out right now till a vaccine like Steve described comes on board, or other antivirals come to fruition. But I think that by simply following basic rules of hygiene and keeping a certain amount of social distancing in place, I think we can really fight this virus at a reasonably good level.
Well, thank you, Madhav, for that answer, and that's very helpful for a broader public to understand. Although the promising, therapeutics, if you get really sick, and possibly eventually avoiding the whole idea with the vaccine is promising. But I think for immediate purposes, thank you for your advice. I want to move, since you talked about the role of testing, I want to ask, there's a question, not me, this was a question that was asked. Two-part question, Amy. This is the first is when we're fully ramped up, what's the rate of testing that you can do? How many tests per day? That type of type of information. And then this is a curiosity question. What is the accuracy of this testing? Do you know that you had false positives, false negatives, and that type of thing?
Yeah. So I don't know if they can let me back on the video. I'd be happy to be, but so first question, how many tests can we do a day? So the governor has put out a proposal that Virginia should be doing 10,000 tests a day. There is the need to do, as we do modeling and as we do testing and we do some asymptomatic surveillance, probably along with symptomatic testing and then contact tracing of people, we're going to need to have a lot of tests. There's 8.5 million people in the state. And there's probably some percentage of those people that need to be tested so that we understand the epidemiology. Having said that, just to have testing available to those who are symptomatic, so far, we've not even had the testing capacity to do that in the state. Just to be clear, the public health labs in the state have only been able to support about 450 tests a day maximum. And here at UVA, we're doing about 600 a day is our capacity. And so we are doing a lot of testing for the Virginia Department of Health, and we see ourselves as a reference lab for VDH, or the Virginia Department of Health, and doing testing in the Eastern Shore or the prison population or vulnerable communities around the state. And so those are our testing capacities currently. If it wasn't for allocation, we would be doing, if we could get the platforms that we wanted, that we've ordered, that we've been waiting for, we'd be doing 3,000 tests a day in our current footprint in the lab. And just to put this all in perspective, I was trying to get some numbers on like what's the most we do in microbiology. And That's urine cultures, and we don't do more than 100 of those a day. So talking about doing 3,000 tests that are biohazard specimens a day it's been altering of the clinical micro operations. And then just to quickly, but we think we can do that within our current footprint and within our current processes, but it's going to stretch the system. The last thing is how accurate is this test? I get this question a lot. Basically, because the FDA has sort of, a lot of these things rushed to approval, there are, by my count, 45 PCR or molecular diagnostic tests that are on the market that have FDA, EUA. They are not all equal. We've actually evaluated a platform and sent it back. And then we had another platform that we said, you know what, you keep it, because they're not sensitive and they don't work the same. And so some of these new technologies, it may seem really great. It's just, you know, buyer beware, because you've got to really refine that. And what we don't want to have is not a sensitive test to say somebody doesn't have the virus. And so right now, all of our platforms detect down to about 100 viral particles per mil. And so those are kind of the most sensitive tests on the market or that are going and cleared by the FDA. So quite sensitive.
Oh, that's wonderful. That's very reassuring to know.
Yeah.
Thank you. And moving to Patrick, yeah, the question is, what is the significance of the results that you got with Remdesivir trial towards the prospects of moving towards a targeted COVID-19 drug? I know you're trying to now do a combination of drugs and so on that's going to build. So the significance of your finding towards accelerating such a discovery, what are the prospects?
Yeah, so it's incredibly difficult to make antiviral therapies in general. We have so many fewer antiviral drugs in the world than we have antibacterial drugs, just because of the way that viruses kind of hijack natural cellular processes. Just means you have fewer potential targets. So finding an antiviral drug that works in the midst of a pandemic is, you know, I think a really impressive sort of thing to happen. In terms of the therapeutic effect of remdesivir, I've been describing it as a solid hit, but not a home run. The difference in time to recovery is meaningful, I think, for patients. If there is a difference in mortality, then obviously that's very meaningful. But simply reducing the amount of time someone is in the hospital doesn't solve this pandemic. It doesn't get us away from having to do social distancing. It doesn't get us away from the need for a vaccine. But it does kind of move things in the right direction. I think the first steps are maximizing the benefit we get out of remdesivir. So one of the questions is which patients benefit most from this drug? What is it better to give this drug early in disease course, as one would expect, versus later in disease course? Can we identify the folks that are really going to get the maximum benefit? And can we use this drug, as we were talking about, in combination with other medications to get even more juice out of it, which is why we're moving on to the remdesivir plus baricitinib study. Outside of remdesivir-based therapeutics, I think if I were putting down my money, which I'm not, would be on convalescent plasma and antibody-based therapeutics. I think are probably the most promising. And one of the nice things about doing the sort of research that Dr. Zeichner is doing is that if you find antibody responses, you know, that are effective in preventing disease, is that then you can turn those into monoclonal antibody therapies and kind of go into a therapeutic direction instead of a purely vaccine direction. So I think the combination of looking at the convalescent plasma, as well as some other folks who are working on kind of vaccine directed studies may kind of give us the next generation of therapeutics that could be even more effective than remdesivir.
Excellent, thank you. Thank you so much. That leads me to the last question for Dr. Zeichner. We are all, you know, eventually the buck stops with you. Once you come up with the vaccine, this is over. So we are, there's a great deal of anxiety and questions and hope. This is the question: how far are we from finding the vaccine, and what are the challenges?
Yeah, my goal is, in fact, to put everybody else out of business. Sorry, Patrick. But I mean, all joking aside, I mean, Patrick and I actually talk a lot, and Amy and I run into each other in the hall all the time. And I just, and that's one of the great things about UVA is it's so collaborative and so many people can work together to answer your question, I mean, Dr. Fauci has said 12 to 18 months. That would be, you know, there are 200 different candidate plus vaccines that people are working on. I think that's great because I think the more shots on goal you have, the more chance you have of scoring. 12 to 18 months would be extremely fast for a vaccine. Previous vaccines have taken a decade or more to go from concept to approved vaccine, but obviously we don't want that to happen here. I think that's a reasonable, time frame, but if it's stretched out longer, I wouldn't be surprised. We're hoping, of course, that what we're coming up with would be a lot faster. And I think our emphasis on trying to make something that you can build fast, make fast for not much money using existing manufacturing capability, because having a vaccine that is safe and effective is one thing. Making 7 billion doses of it and distributing it around the world may be even a bigger challenge than the immunological challenge. So we're hopeful that that may be a good strategy.
Yeah, thank you, Dr. Zeichner, for that. In fact, what you're describing in my mind, it translates to use inspired. I mean, you can go at the fundamental basic science discovery to end up on a vaccine. But what is interesting is you're throwing in the constraints of manufacturing scalability so that this will actually make it. And once you have that enterprise in mind and the mechanisms that you can actually scale up in manufacturing, that really leads you to a unique path of things that you would try, and if successful, then the benefits are immediate. I mean, I applaud you for an approach that you're taking that's so novel, and thank you for that. So with that, I think we're running out of time. We're over time. Let me wrap up by saying a few things. First of all, the University of Virginia, has been pursuing basic research and leading discoveries, and that has brought prosperity, health, and national security in many different fields. But by focusing our resources and expertise in the near term to tackle this pandemic, our researchers are living up to the vision for UEA to be both a great and a good university, and serving in the interest of the public good. I mean, this is, I couldn't bring more examples than this to make that point as to how we are thinking here as a university. And what we have shared today is our current approaches and ideas that we have brought to bear and continue to pursue. As we continue to pursue our research, as Patrick said, we will produce more knowledge, and the new discoveries will come, and that will accelerate our conquering this pandemic. So I want to thank all our researchers for all their hard work. A special thanks to Madhav, Amy, Patrick, and Stephen for taking the time today in the middle of their busy schedule to explain their research to all of us and answer some of these questions. Finally, I want to thank all of you for joining today. And that concludes this panel, and the panel is now